Melissa Camillar Lopez.
And I'm Sandra T. Lucas
And dito kami para i-report yung article naming napili.
for finals.
The title of the article is Thermodynamics of Interaction of the Food Additive Tartrazine with Serum Albumins, a Microcalorimetric Investigation, journal article of
Anirban Basu.
and Gopinatha Suresh Kumar.
Sa article, yung tartrazine as orange-colored, water-soluble monoisopyrazolone dye. And then, it is commonly known as FD&C yellow number 5 and food yellow number 4. Tartrazine was commonly used as food colorant that can be found in soft drinks, for example, Mountain Dew, and then juices, cookies, ice cream, sauces, mustard, and snacks. Also, it was stated that tartrazine has synthetic azo dyes containing NN linkages and aromatic rings, which is known to be hazardous to human beings, and it has effects to the children, which is hyperactivity. According to ADI, the acceptable daily intake of tartrazine for human beings is 7.5 milligrams per kilograms. Serum albumins are the most abundant transport proteins in blood plasma that perform multiple physiological functions and some biological functions such as membrane penetration, metabolism, half-life, influence several other pharmacokinetic properties. The article used these two types of serum albumin. First is the human serum albumin, which can be found in humans from our liver. And the second is the bovine serum albumin, which can be found in cows. One of serum albumin is the strong binding ability sa tartrazine. Ibig sabihin, kinokontrol niya yung toxic effect ng tartrazine. Ayon sa mga researchers, wala pang nakapag-perform ng evaluation sa thermodynamic properties ng human serum albumin at bovine serum albumin using microcalorimetric techniques.
So therefore, this study presents a detailed evaluation of the energetics of the complexation of tartrazine with two serum proteins using microcalorimetric techniques. So they start the evaluation by first setting the controls of experiments using fixed volume of tartrazine solution. So in this case, they use 10 microliter of tartrazine solution. So they start the setup. So from the stirring syringe kung saan nakalagay yung tartrazine solution with fixed amount of 10 microliter. So here dito siya. So may two identical cells, yung reference cells sa yung sample cells. Yung apparatus, yung whole apparatus is thermal conducting with adiabatic jacket. So para maging heat insulated siya. So merong temperature detector that detects the temperature difference between the reference cell and the sample cell. So yung reference cell usually filled with buffer siya or ng H2O. Then yung sample cell, doon naka-contain yung protein natin na serum, yung dalawa, yung BSA saka yung HSA. So constant power is applied to the reference cell kasi nagda-direct siya ng feedback circuit. Tapos ina-activate niya itong heater located sa sample cell. So may time dependent input siya para ma-maintain yung equal temperature between sample and reference cell kasi kailangan yung delta T nila is approximately 0 kasi dapat approximately equal sila. So exothermic siya pag yung temperature cell nag-increase upon addition ng ligand. Feedback power of the sample cell nagde-decrease. So yung feedback power niya dito magde-decrease pag nag-increase tayo ng temperature sa sample cell. So ito yung model na ginamit dun sa experiment, yung MicroCal VPITC. So as we can see here, meron na siyang software kung saan ma-visualize natin yung graphic representation ng experiment habang ginagawa na nila. So here, ito yung mga controls ng experiment. Meron tayong duration time of each injection na 10 seconds, thus yung delay time natin is 240. So from here dito sa setup, Yun sa steering syringe, nandun yung ligand natin, yung tartrazine. So, pag finil natin siya or pag in-inject natin siya dun sa may sample cell, meron duration time na 10 seconds. Yung 10 microliter na volume ng tartrazine. So, after natin mag-inject, magkakaroon ng duration time na 240 seconds after mag-inject ng second. So, ito yung nangyayari habang pinoproceed natin yung experiment na yun. So, ito yung syringe. Yung volume na, yung amount na nandito is yung 10 liter na tartrazine. So, dito, ito yung, bale yung kanina, ito yung sample cell natin. So, ngayon, yung first injection is made all injected ligand is bound to target macromolecule. So, yung sa first injection natin, magbabound lahat ng protein natin, yung serum. So, ayun. So, exothermic reaction siya kasi dito yung baseline natin. So, upon injection ng tartrazine, bababa yung power niya. Itong power na to, ito yung galing dun sa power na dinana natin dun sa reference cell. So, bumaba siya kasi nga exothermic siya. Then, dito, di ba, yung kanina, yung first na finil natin is ang duration time ng 10 seconds. Then, after nun, magkakaroon tayo ng delayed time na 240. Ang nangyayari dun sa delayed time na yun, yung heat, babalik siya dun sa baseline. Babalik siya dun ulit sa baseline. So, yun yung purpose niya kung bakit tayo may delayed time interval na yun. So, ito yung sa second injection naman. So, bababa siya ulit. Pero hindi na siya same dun sa una. Mababawasan na siya. After nung second injection, babalik ulit siya dun sa baseline. So, ito yung series nung maraming injection. So, itong region na to, ito yung delta H. So, nag-proceed lang yung experiment nila until wala ng delta H, ma-reach na yung pinaka-baseline. Dun sa experiment, ang pinaka-masusupply na data itong delta H, yung region dito, saka yung KB, yung binding constant, which is yung parang average. Magsaset ka ng tangent line dyan, tapos i-average mo. Yung pinaka-average niya yung binding constant. At yung binding constant equal naman siya sa 1 over KD. Yung KD, yung equilibrium constant, which is kailangan natin para ma-evaluate yung other thermodynamic properties such as yung Gibbs energy. Tapos itong N is stoichiometry. Ito yung number ng binding sites. For example, familiar tayo sa EDTA, which is may 6 binding sites siya. So, ang tawag sa kanya hexadentate. So, yun yung minemeasure ng N. So, ito yung pinaka-graphical results ng kanila experiment. As we can see, masyado sa graph, base doon sa graph, makikita natin na mataas yung affinity ng both protein natin dun sa tartrazine. Kasi wala siyang laging phase. As in, diretso na siya from pinakamababa, pataas lang siya, hindi siya naglalag ng values. Mamaya, makikita niyo yung figure. So, ito yung mga figures. So, ito yung example nung nakuha nila sa experiment kanina. So, tatlo kasi yung kinds nung parang graph na maipe-present. So, ito yung parang, familiar tayo dito, yung sigmoidal curve. So, from dito, sa pinaka-lower part, is parang may lagging phase siya bago siya tumaas. So, ibig sabihin, yung concentration na kailangan natin dyan is mataas bago sila mag-bind or bago niya ma-attain yung affinity. So, dito naman, naglag siya, then suddenly, biglang taas. So, ganun din. Nagre-require siya ng mataas, high concentration. Pero yung kanina, at lower concentration ng protein, yung initial amount ng tartrazine, which is mababa lang din, is lahat yun is nag-bind agad-agad. So, hindi kailangan ng high amount ng protein para maging capable siya mag-bind kasi at low concentration, kaya niya na. So, ito yung ano nun, yung protein, concentration ng protein over KD less than 2:1. So, negative, so, nasa 0 point something siya kasi yung protein mo is mababa. Hindi siya ganun kataas to require the affinity na kailangan natin.
So, at 298.15K, mas mataas yung binding site binding affinity ng bovine serum albumin. Ayan, nakita naman natin is 1.92 plus minus 0.05 times 10 to the fifth per molal. So, mas mataas siya sa HSA. So, itong N values, siya yung number of binding sites. So, yan, tignan mo, yung BSA tsaka HSA, nakita naman natin na malapit siya sa 1. So, binding size nila is 1. So, yung KB, siya yung binding constant. So, yung binding constant, diba kanina na-supply na siya nung ginawang experiment. So, yan, yung KD equal siya sa 1 over KD, which is equal din sa concentration ng ligand times concentration ng serum over concentration ng ligand plus serum. So, ayan, nakuha na natin yung equilibrium tsaka binding constant. Then, dito sa third equation, yung delta G, makukuha natin siya kasi itong dalawa constant naman to, yung isa constant din, yung temperature given din yan. And then, ito nakuha na natin dito from dyan. And then, yan, nakuha natin yung delta G. Then, next is, makukuha natin dito is delta S because itong delta G is nakuha na natin. And then, yung delta H, diba kanina nakuha din natin siya based sa experiment. Then, ayan, makukuha natin yung delta S.
Kaninang delta H, siya yung na-supply dun sa data, siya yung nakuha, na-obtained experimentally, yun dun sa may region kanina. So, yung T, constant yan, constant yung gagamitin natin kasi controlled siya. Kasi dalawa yung set ng experiment na ginamit nila. First is yung controlled, yung mga parameters. May mga parameters sila na sinet na constant, tulad ng volume sa kanilang temperature. Kasi mamaya, yung sa second phase ng kanilang experiment, magpe-perform sila ng differential scanning calorimetry kung saan i-vary na nila yung value ng temperature at saka ng ibang parameters. So, at fixed temperature, yung 298.15 na sinat natin, ang nakuhang delta RH, yung enthalpy, sa BSA at saka HSA is parehas na negative. So, ang enthalpy is, di ba pag negative yung enthalpy, ibig sabihin, favorable siya exothermically. So, ibig sabihin, heat releasing siya, hindi siya nag-absorb. Then, kanina, nakuha din natin yung T delta RS or yung entropy. So, sa BSA, makikita nyo, positive siya. Sa HSA, negative siya. So, ang conclusion nila dyan is sa BSA kaya siya positive kasi mas favorable yan. Di ba remember ang entropy is yung disorderliness. So, ano ba ang natural tendency ng lahat ng reaction? Di ba disorderliness? So, pag positive siya, mas favorable kaysa dito sa negative. Dito naman sa result ng delta RG o yung Gibbs free energy. So, sa BSA at saka sa HSA, ayan, para sila negative. Ang Gibbs free energy, di ba, pag negative, ibig sabihin is spontaneous siya. So, meaning to say, both reaction nila with tartrazine is spontaneous.
Next is differential scanning calorimetry. So, dito, ito yung salt-dependent ITC studies. So, dito, nag-a-add ng concentration ng sodium. So, nakikita nyo naman na kung nag-a-add ng sodium, is bumababa yung binding constant, which is nagle-less yung affinity nung serum nitong BSA or kaya ng HSA. Nagle-less yung affinity niya kasi dahil nag-interfere yung counter ion. So, ito yung relationship ng K versus salt concentration, where delta Rn is equals to the number of ions released upon binding of ligand, and the Sa is the charge of the bound ligand, and the Phi is the fraction of Na plus bound per ion. Anion.
So, here yung expression, pinapakita niya lang yung relationship ng K at saka sa yung concentration ng Na+. Kanina din sa data nakita natin, ayan, yung Na+, nagdadagdag siya ng 10 nung concentration. So, for every dagdag niya is bumababa yung K. So, ayan, kasi yung inversely proportional relationship nila.
Yung graphical results ng nung data kanina. So, 'di ba sabi nga kanina yung log, yung K tsaka NA is inversely proportional. So, yung slope nila is downward slope. So, ayan, yung mas mataas yung mas mataas yung nakuha sa BSA. So, ibig sabihin, mas sharper yung variation.
Ayun. So, mataas yung value ng BSA pero negative siya kasi nga downward slope. So, ibig sabihin, at certain amount na dinadagdag natin na concentration ng Na+, mas mabilis na nagbabago or naapektuhan yung equilibrium constant ng BSA compared sa HSA. The delta RG can be divided into two contributions. So, first is yung polyelectrolytic and non-polyelectrolytic contribution. So, yung delta RG polyelectrolytic contribution gives energy can be calculated using this equation. So, ito yung Gibbs energy contribution coming from coupled polyelectrolytic force. Example niya is yung condensed counter-ion. Yung isa is the standard molar Gibbs energy contribution coming from non-polyelectrolytic sources. Example niya yung hydrogen bonding, van der Waals interaction, saka hydrophobic transfer. O, yung isang ano natin, yung temperature-dependent calorimetric binding studies. So, ang ginamit nilang temperature interval is between 283.15 to 310.15 Kelvin. So, dito, as we can see, pataas ang pataas yung temperature na sinusupply nila. Then, ang effect dun sa binding constant is bumababa. Meaning to say, pag ini-increase din yung temperature nila, yung affinity nagde-decrease. So, kung mapapansin nyo dito sa HSA, kung mapapansin nyo, yung T delta S o yung entropy, favorable lang siya sa 283.15. From 293.15 to 310, negative na siya. So, dito sa data, kinuha rin nila yung heat capacity, yung standard molar heat capacity. Ito yun.